paper is entitled Biocultural Collections, Quo Vadis.
Good morning, everyone, and thank you for being here. My name is Kenneth Reginald Otero Walker, and I'm the project manager at the New York Botanical Garden on a project to secure, digitize, and increase access to the biocultural collections currently held in the Harding Lab of the Institute of Economic Botany. Through the NSF grant provided, we hope to preserve, but through the grant provided and the curation of 11,000 objects, as well as 23,000 common names, we hope to preserve traditional knowledge for use in ecological problems, to support local livelihoods, and also to help support food sovereignty. Biocultural collections have been important to the collections at the New York Botanical Garden since the early collaborations of famed American botanists John Torrey and Edward Palmer. As you can see there at the bottom, this is a letter from Edward Palmer to John Torrey, asking him to name and describe some of the plants collected on his expeditions into the Midwest and Mexico. These collections were advanced by the first curator and co-founder of the New York Botanical Garden, Henry Herd Rusby, whose research into plants such as cinchona helped place the New York Botanical Garden firmly within the scientific community. The next step in this collection process happened with the founding of the Institute of Economic Botany. Well, the founding of the Journal of the Economic Botany, first with Edmund Fulling and Henry Allen Gleason. And then the next step was obviously the Institute of Economic Botany. The Institute of Economic Botany was co-founded in 1985 through the collaborations of Gillian Tomlinson Prance and Michael Jeffrey Balick. And since its inception, it is focused on the interrelationship between linguistic diversity, biological diversity, and cultural diversity, and the use of that research in conservation. The IAB has also produced significant research in ecosystem valuation, functional food medicines, the stewardship of agricultural diversity, and crop wild relatives. Its curators have sought to preserve ethnomedical and ethnobotanical traditions and have linked and networked with local stakeholders in initiatives of value added within value chains of non-timber forest products. Its curators have also explored the role of access to healthcare on ethnomedical traditions of immigrants within the city of New York. So the IAB houses several very important collections, among them the Rusby collection, the projects of plants and people of Micronesia and Oceania, as well as the Caribbean and Latino ethnobotany and ethnomedicine program. In total, there are over 10,000 specimens with which this project deals with. These collections have been advanced by prominent ethnobotanists such as Prance, Balick, Bennett, Boom, Ballet, Daly, Paddock, De Jong, and Peters. And currently they continue to be advanced by collectors Balick, Vandebroek, McAlvey, and Vardaman. So in moving these collections, it's very much a labor of love, because not only do these collections contain the specimen of its curators, but also of their research associates, as well as many postdocs and students who have worked at the IAB since the 1980s. Though the IAB has been a center for ethnobotanical research, it was never designed to house these collections. Furthermore, the IAB is slated for destruction within the next 2 years. As these collections have stood separated from the general herbarium, they don't participate within the added benefits provided by the herbarium, such as temperature and humidity control, as well as an integrated pest management strategy. In considering the challenges of moving these biocultural collections into the herbarium, there was special attention paid to the fact that regular botanists and systematists don't usually appreciate the non-fertile material, as well as the many sterile duplicates that are produced through ethnobotanical research. Similarly, the non-standard size of reference specimens from botanicas and other market vouchers, as well as cultural artifacts, present increasing problems of storage and space for the herbarium. Digitization of biocultural collections begins first with EMU, which is a proprietary software used by museum collections in which label information and taxonomy is recorded. After that minimum of information is recorded, these collections, which have been usually stored in newspaper, are sent to mounting room, where they can be mounted to herbarium sheets of paper. After being mounted on herbarium sheets of paper, they then go to the digitizing lab, which can be seen in the upper right-hand corner, and their pictures are added to their digital files. Once there is a complete file, these specimens can be loaded onto the virtual herbarium expedition platform, which is managed through, which links with data aggregators such as iDigBio, and is managed through Digivolve, so that these specimens can be transcribed. Dealing with these collections causes us to consider special ethical questions. First among them are Nagoya Protocol and CBD, the Convention of Biological Diversity. We are required as researchers to provide free access and to share the results of our research with the scientific community. In order to do this, however, we have to have adequate documentation of prior informed consents, as well as be in constant dialogue with communities regarding their rights over their intellectual property. The solutions to these considerations are appropriate collection management, which is constantly analyzing these collections through conversations with curators, as well as conversations with communities. All of the specimens generated through this project are shared. However, through the use of a blur tool, information which needs to be protected can be blanked out in the same way that location of endangered species is protected within the herbarium. These biocultural collections within herbarium play an unprecedented role in science. Through GIS and conservation, we have been able to do unimaginable things in terms of plants like sacred white lotus of the Himalayas. Similarly, collections such as the historic Henry Hurd Rusby pharmaceutical collections have been used in the DNA barcoding of dietary supplements such as black cohosh, showing that these specimens are usually pharmacologically active hundreds of years after they've been collected. So also within the herbarium, there are several platforms which advance the reach of herbarium specimens. The first that I want to mention is biotic interactions, which allows us to examine the interrelationship between specimens and their biotic environment, both human and non-human. Similarly, there's a platform called the Hand Lens, which allows curators as well as researchers to explore specimen in new light, telling the personal sides of stories which specimen may represent. through collaborations and interdisciplinary work with the humanities department, these specimens and the stories that they tell can begin to reach new communities and new audiences. The herbarium has also begun to link with artists to bring these specimens outside of the herbarium and into the garden at large. Here is pictured a specimen from the gallery of Sam Ankin from an exhibit called Re-Orcharding New York, in which herbarium specimens from the herbarium were used to show the biological diversity of crops that once existed in New York City. Similarly, Jamaican-born artist Ebony G. Patterson has recently used extinct species within the herbarium and created glass sculptures and brought them into the conservatory in order to help people understand what it's like to live with the ghost of an extinct species. These biocultural collections have also been extremely useful to the herbarium itself in its programs of educational and public outreach. Suggestions? These biocultural collections are allowing traditional peoples all around the world to have access, not only to this knowledge, but to these spaces. In order to continue this movement, it requires that emerging ethnobotanists are fully integrated, not only within the scientific community, but also within the communities that they research and represent. It is important that collections reflect the attitudes and the ideas of community members. And in that vein, one of the next steps forward must include labels and data sovereignty for traditional and local peoples. Similarly, there should be more focus on access and benefit sharing and how we trace specimens and the benefits that have been produced from them in scientific research. The New York Botanical Garden strives to operate well within care and fair principles of scientific data and also cultural and data sovereignty. But in that vein, it also seeks to be at the forefront of ethnobotanical research. And so there must be some changes made to the economic botany data standard or to the competencies that allow us to exchange research so that this important research can continue into the future. Thank you. And I would also like to thank Siri Sylvia Patricia Reese, who is one of the first major ethnobotanists to do a major exploration of the biocultural specimen held within the New York Botanical Garden, and whose research is invaluable to many of us here. Comments, concerns, questions?
The community can access the data or somehow make use of this collection, that it's not only accessible or usable by professionals. Is there a way to kind of involve community project or citizen science or greater community?
The answer is yes. Through our CV Star virtual herbarium, all of the community can access all of the records that are housed and digitized within the New York Botanical Garden. The information which is blurred can also be requested from collection managers for purposes of research. Wendy?
Might need to be patriated under ag crop, might actually call upon you to take the data.
Yes, that is a constant consideration. And so we're constantly looking and trying to determine the provenance of cultural artifacts as they present themselves. And should anything that is questionable prop up, then repatriation will be our first. Yeah.
Question about your blurring of the data. Herbarium specimens are often collected in multiple duplicates. So you're really only in control of specimens that are held by your institution. How are you dealing with, this problem of duplicates exposing data on another system?
Well, you've identified, one crucial problem, and that requires the interconnection of these various botanical research institutions. And so while I can blur, these documents here at the New York Botanical Garden, as you so, correctly stated, those duplicates or even, the specimen which are sent to, say, the Smithsonian have not been blurred and so still represent a weak spot within the protection of this information. Anyone else?
Thank you very much for the presentation and for everybody. Can you actually go back to your thank you slide? It was so beautiful and it went so quick. So
Sorry.
on behalf of everybody.